styrene, butadiene, rubber, copolymer, how we can fine tune their mechanical properties, maybe of modulus or elasticity. It's very clear right now, right?
Yes, sir.
Yeah, fine. The same concept applies for other copolymers. Say, for example, if you see the high impact polystyrene, if you see the chemical composition, I mean, what are their combination? Like they are made up of polystyrene based plastic as well as the combination of polybutadiene. So if you see the polystyrene based plastic, I mean, the mechanical properties, they are highly brittle. Okay. So to modify their brittle, I mean, highly brittle nature, they used to embed some sort of little bit of rubber molecules. Okay, it's based on polybutadiene. So once you do that, the material, I mean, polystyrene based things product can be flexible as well as having a very high strength. Okay, I mean modulus. Okay. So today we'll see little about protein structure and its basics and their types like a three component protein, etc. And then we will get into the nanoscience and technology. Okay. So protein, everybody knows. I mean, protein is very important, and if we see the definition of protein, it's made up of, it's exactly polymer and it is made up of several small units, which is nothing but amino acids. So the many small number of amino acids are combined and together and it forms macromolecule or the protein structures. Okay. So proteins do not have a uniform structure. That means they do have primary structures. But if you see the, kind of secondary structure, how exactly they orient or they get, confirmation or the configuration. There's no like a defined structures. Okay, due to the amino acids and their, structures like a formation of polypeptide bonds, etc. And nevertheless, they do have a major structures like four different folding structures according to the protein folding phenomena, like primary structures. That is nothing but the amino acid. Like what are the amino acids you're, taking, or the sequences of their amino acids. Okay, and the secondary bonding means like. What is the interaction? Like secondary interactions, say for example, hydrogen bond between the peptide bonds. Okay, and then a tertiary structure means like if there is interaction like ionic or covalent or hydrogen bond or hydrophobic, when the protein gets interact with the water or any other organic solvents, etc. Okay. And again, quaternary structure that involves again kind of self-assembled structures, and it's nothing but the formation of more number of small subunits. Okay, just like tertiary structure, but combined with the tertiary structure, it also has some sort of subunits. Like while forming those aggregates, it will form, as say, either a nanorod or some sort of flakes or some sort of spherical structures, etc. Okay. So this is how the overall structure. So left hand side you see here, this is the primary structures. Okay, the each, the bubbles or the sphere looks, they are corresponding to the single amino acid molecules, and many a small number of amino acids combines and it forms a macromolecule or the protein. Okay, and all these things are like secondary, tertiary, and quaternary. Okay, secondary structure. If you see the two types of secondary protein structure, they have a alpha helix and beta sheet. Okay. Okay, alpha helix means it's you can look here, it looks in the form of spring structures. Okay, and beta sheet means the chains are oriented in such a way that, zigzag manner. Okay, zigzag means the chains, they align in a perpendicular or the parallel way. Okay. So how it happens? Okay, the either beta sheet or alpha helix. It is due to the formation of weak hydrogen bonds into one or more structural form. Because if you see the primary structure of your protein, they have a different amino acid. That means every acid and in between that you have a peptide bonds. On top of that, there are changes in the charges in the main backbone. Okay, like they do have a N terminus and C terminus, etc. Okay. So once they interact with some solvent or water, what will happen? They'll start to assemble, and as a result, the due to that variations in the charges, they start to assemble according to the the secondary bonding or the interactions like hydrogen bond formation. As a result, they'll start to align either in the form of alpha helix or beta sheet. That depends on your primary structure or the amino acid, whatever the selection of those. Okay? And the tertiary structure means it's a combination of both actually, like kind of you have a beta sheet combined with that alpha helix structures. Okay, that occurs usually like when the protein gets interact with the either organic solvent or kind of water molecule, etc. They'll start to assemble. As a result, what will happen? They will interact with the water molecules and these to form some sort of aggregate structures. So that is called tertiary structures. Again, the quaternary protein structure means a protein consisting of more number of one amino acid chains and each participating in the final 3D shapes. That means you have certain like kind of uh, alpha helix and beta sheet, they used to, aggregate or they used to form one the parallel to each other, each one. And there are sometimes they used to form, opposite to that of one to other direction, like beta sheet and opposite to that of alpha helix formation. So those kind of different subunit formation will happen in the coronary protein structures. So this secondary structure can be visualized either by using spectroscopy or kind of light scattering technique, because if you pass the light, either UV or any other laser light, what will happen? The the size of this aggregate or the formation of this structure will be varied as compared to different classification like a tertiary or secondary or the quaternary. So more closely now you're gonna study more into the chemistry aspect, like primary structure and how the peptide bond is forming and what is N terminus and what is C terminus, etc. Okay? And this AAS means amino acid sequences. Okay, primary structure is a protein refers to its amino acid sequences, and usually in amino acid in peptides and proteins typically of minimum 13 amino acid sequences should be there, typically of 200 to 1000 repeating unit at the amino acid sequences, and they are combined together by peptide bonds. Okay, you can see here there is a carboxyl group and then amine group. Okay, and they react and then the peptide bond will form. Okay, and the usually the peptide bonds a formation of peptide bonds between the carboxyl and amino groups of adjacent amino acids, and the backbone of all protein consisting of N terminus as well as the C terminus. Okay, if you see the charges, N has positive and C has negatively charged. Okay, and yeah, that also regulate those formation of structures like a secondary or tertiary or a quaternary. Okay, and the average yeast protein contains 460 amino acids. Okay, because of the average molecular weight of amino acid is 113 daltons, or 1.13 kilodalton, and most of the protein exhibit the molecular weight of 50,000 kilodalton, sorry, 50,000 dalton. Okay, and this is how exactly it forms the secondary alpha helix structures. Okay, uh, usually it forms, like very very short range period of folding elements, and this includes the helix and the beta sheet, and sometimes they turns into the spiral form also. In the helix, as shown in the figure, the backbone cylindrical spiral structure in which there are 3.6 amino acid sequences per turn can be seen. Okay, and yeah, the R groups point out the helix and the mediate contact to other structural elements in the folded proteins. Okay, and the alpha helix, is stabilized by the hydrogen bond between, the carbonyl groups as well as the amide from the nitrogen atoms. Okay, and that are oriented parallel to the helix axis. That is how the alpha helix structure is forming. Okay, and the hydrogen bond occurs between the residues located in the N and the N+4 position, led to the top one to another. Okay? And it is not the, kind of ideal. It depends on the types of amino acids present in each proteins as well. Now you see the secondary structures. How the beta sheet formation in a beta sheet, like each beta sheet strands adopts an extended conformation. Okay, and they tends to occur in pairs or multiple copies. Of course, yeah, it depends on the water or how we are taking the, the processing of those protein. Okay, either the organic solvent or the water, etc. And usually the beta sheet formation that interact with one another via hydrogen bonds. Okay, directed perpendicular to the axis of each strand. And if we see the, top view, this is how they looks. Ideally, the chains looks very symmetrical way, and the strands can orient anti-parallel. Sometimes they can, orient in the anti-parallel way as well. Okay, and R groups of every other amino acid point up or down related to that of sheet, and yeah. Minimum, most beta sheet stands in proteins are 5 to 8 amino acid sequences long. Okay, and beta sheet again, they can turn into the consisting of 3 to 4 amino acid groups. Okay, and if we see the property wise, like why this kind of like why to study about this beta sheet or the secondary different secondary structures, etc. Because when it comes to the protein properties, these things are matters. Okay, of course primary structure is very important, okay, that defines technical properties, either mechanical or other biological characteristics. But yeah, we see the mechanical properties more closely, like beta sheet provide more crystallinity to the material, the protein. Okay, so if you want to fabricate either film or some sort of biological construct like a hydrogel or scaffold, so you should have those kind of significant mechanical stability, so that the tissues can regenerate nicely, without hampering of those native tissues or the organ. So if we see the alpha helix and beta sheet, people prefer to have more of beta sheet content once protein is being processed. Okay. So more the beta sheet content, you will have more mechanical strength or the properties. Okay. So this is again the tertiary structure. So you can see here folding, unfolding. You have a primary structure. Once they interact with some either water or some sort of organic solvent, they'll start to fold. Okay. So once it is folded, you can see the core will be of either maybe a protein and maybe the surface, and then on top of that your water is to get accumulated. Okay, so this is how the protein once it is folded or once it is aggregated. Okay, it looks it forms like this. Okay, that formation could be of like a backbone trace or sometimes they used to call ball and stick. How to see them. So sometimes they used to be in the form of like a ribbons and combined with this alpha helix and beta sheet, either maybe a parallel or anti-parallel way. So basically, how this aggregate is happening, because you have protein different charges, and on top of that, that solvent or the water, they have different charges. So once you mix them, they may dissolve or they may not dissolve. Okay? And there'll be some, the saturation limit. Once the saturation limit exits, they will not dissolve at all. As a result, they'll start to fold. That means they'll start to aggregate, and then they used to have, form these kind of structures. Yeah. So tertiary structure refers to the folded 3D structure of protein, and it is also known as the native structure or the active conformation. And the tertiary structure is mostly stabilized by non-covalent interaction. Yeah, all these things are non-covalent interactions. That means if we heat them slightly above the room temperature, they'll start to get away. Okay. And the folding of protein is thought to be driven by the need of place to the most hydrophobic regions in the interior, out of contact with water or any other organic medium. And yeah, this can be visualized by using modern X-ray crystallography or the solid state NMR, etc. So again, the last one, the quaternary structure, means it is a combination of several subunits. That means you do have some sort of fibrous domain or the nanorod domains, as well as the globular domain. That means some sort of aggregate, the form of spherical shapes, etc. And yeah, multi-subunit or multimeric proteins have another level of structural organization known as quaternary structure of proteins. And it refers to the number of subunits and their relative position and contacts between the individual monomers. And of course, this also happens once once they start to interact with any kind of media or the solvent. Okay, media means, see, look, proteins always interact with different media, in our body, right? Either could be of blood or water or any kind of buffer solution, right? So one should know like how the protein structure changes when they interact with the different media or the materials. So now here, this is one best example of protein. That is based on hemagglutinin based surface protein, and basically it's a influenza virus. And yeah, you can see here there the formation of different multi subunits. In the form of fibrous domain as well as globular domain. Okay, so if we see the applications, proteins are mainly used for biomedical sectors, like can be used for tissue regeneration as well as drug delivery sectors, as well as for wound regeneration or the wound dressings, as well as can be used for biosensors, etc. Okay, so we do have some other concept kind of, called recombinant protein. Okay, it's nothing but, okay, recombinant protein is a manipulated form of protein. That means it is prepared in our laboratory. Okay, and so by the way, proteins can be fabricated as, isolated from the animals as well as the insect as well. Right? Proteins can be obtained from the natural resources, maybe of animal or some planet, etc. All, right? So I'm not going into that area, uh, but yeah, nevertheless, you try to have this kind of knowledge, like what is recombinant proteins. What is this? This may be new for you. Okay, so that means this recombinant protein means you are going to fabricate the protein in the laboratory. Okay, that means you are identifying some of the protein sequences, sorry, amino acid sequences from some protein, and then you are going to design those protein in the laboratory by using for biofermentation technique. Okay, so here you can see here, recombinant protein is a manipulated or the tailor-made form of proteins which is generated in various ways to produce large quantities of protein, to modify gene sequences and manufacturing useful commercial products. Say, for example, many people, the research or the recently the spider silk based proteins are being used for, in various purposes. Okay, in fact, this was one of my postdoc work. Like what we did, like in the European forest or the garden, we have selectively chosen Arrhenius diameter based spider, and we have studied the chemical composition of this spider web. Okay, and like what are the amino acid sequences, etc. And then we have seen that, said spider webs are very strong, and yeah, they've got excellent mechanical properties, etc. And on top of that mechanical properties, if we see their biological characteristics, they have got a very less or no toxicity at all. And if you see their immunoreactivity, almost it is 0, and they have got a good biodegradation as well. Okay, so if you see their chemical composition or the structure of that spider web, they do have a kind of structures like core shell structures, and core has lipids, and the sorry, shell has lipids, and the core has kind of different amino acids. That means proteins. Okay, and yeah, and what we thought, why don't we mimic those spider webs in the lab? We can also prepare those proteins in our lab, right? Because one advantage of preparing proteins in the laboratory is that means recombinant protein, you can have the consistent property. Okay, so basically most of the people, they used to isolate the proteins from the animal resources, or kind of from the planet sources. Okay, so once you dependent on natural resources, what will happen? We do have a different session, right? We have summer, we have autumn, spring, etc. So nature changes, keep on. So as a result, the protein where you are isolating, that properties will not be remain, ideal, constant. The properties changes keep on. Because to get rid of those issues, the people go ahead with this kind of biofermentation technique for, synthesizing recombinant protein. So once you do that, the property consistency will be there. Okay? So this was the case, like a recombinant spider cell protein has the molecular weight of 45 kilodalton, And we used to call this one as a C16. Okay, that means it has a repeating units. I mean, 16 repeating units of so amino acid like glycine, thiamine, and adenine, etc. So this is the scheme where I mean people typically use production of recombinant cell protein or any kind of proteins using biocombination techniques. So for our case, first we have obtained the spider's web. I mean the information, genetic information, and then we have prepared the plasmid with those kind of obtained genetic information, and we passed this one to the I mean the E. coli based substrate. Okay, to start the fermentation technique. Once the fermentation is, I mean, completed, I mean, the with the help of this bacteria and the protein will start to grow, and then finally you can, I mean, isolate the proteins or the purify the protein by using different standard techniques. Okay. Once the protein is purified, I mean, can be used either to make a thin film or fiber or foam or hydrogel or particles, non-woven and capsules. Now, so we can start discuss now here also. Like what is fiber and what is thin film and what is foam and what is hydrogel and what is particles? Probably this may be new for you, right? What is fiber first of all? Why to call fiber first of all? Can we prepare fiber by using metals?
Yes, sir.
So what is the difference between metal fibers and metals?
Flexibility and movement freedom. Fibers provide more leeway in
Yeah,
that
that's what.
movement.
Correct. But why? How?
Metal in metal we have more metal bonding, but in fiber we have spun metal wires. Something.
That means the molecules, I mean, the atoms are oriented in a particular way. I mean, the moment when you spun the metals precursors, or when the moment you fabricate the fibers from any sources, either metal or ceramics or polymers, you are pushing the source. I mean, you are spinning the materials at particular fashion. Okay, that means you are trying to reduce the I mean irregular manner. I mean the I mean atoms arrangement. Okay, as a result, what will happen? Your percentage crystallinity will increase, okay, compared to that of other, I mean, solid material or solid metal or the other materials. Okay, so that means fiber has more crystallinity, and because of their, I mean, regular arrangement of those molecules or the atoms. Okay? So as a result, yeah, that's what you get more mechanical strength, and you are sacrificing the elasticity of material. I mean, in case of fibers. And what about the other terms like films? Films, everybody knows thin film, okay? And what is foams? If we introduce during the fabrication, either solution casting or kind of friction stir casting or I mean any other fabrication technology, if we introduce some sort of gases, maybe of some helium gas or any other gases, okay, even oxygen as well. So what will happen? The material, I mean, in case of film, the structure will change actually. I mean, there will be the gases will get into that material, and they will produce some voids into the, I mean, substrate. As a result, your substrate, either in case of film, you will end up with this kind of porous structures. That is called foam. And hydrogel is kind of very like jelly-like material, which has 95 percentage of water and 5% material. Usually, the hydrogel, usually the formation of hydrogel is like material should dissolve in water, and once the water is sorry, once the material is dissolved in water, then hydrogel will start to form. So we'll see all this thing in the, I mean, next class, or I mean the upcoming class. Again, nonwoven means collection of fibers, and it forms a mat membrane or the mat, okay, which is called nonwoven membrane or nonwoven. And capsules, again, like a kind of core cell structures. Core may be of different and shell may be of different. I mean, this all these things are used for different purposes. Okay, say for example, fiber. If you could produce fiber which has high mechanical strength, it can be used for reinforcing other materials, say of polymer or the ceramics. Okay, so we'll stop here, and maybe next class we'll start this nanoscience and technology. If you have any doubt, we can discuss now. I given four seminar topics on this field, like hydrogels and recombinant silk protein, etc. So if you are interested, you can pick up it, and maybe you'll learn a lot of things if you are interested. Okay. Okay, then if there's no doubt, we can sign out. Okay.
Thank you, sir.
Yeah. See you then.